Hello, Dr. Pascual Leone. I wanted to express our gratitude for your participation in our Meet the Scientist, a friendly interview series. This series is part of our Penn Program's Distinguished Lecture Series, abbreviated DLS. I'd like to start off with some general questions. The first being, why did you decide to become a researcher? The second, with your current research, how did you begin the process? And lastly, how did you become interested in this particular niche of your research?
Hello, it is a pleasure to be with you. I'm honored by the invitation. I thought I would start by telling you why I got into science and how I ended up doing what I do. My name is Álvaro Pascual Leone. I'm a professor of neurology at Harvard Medical School, and I am a senior scientist at the Hinda and Arthur Marcus Institute for Aging Research and the medical director of the Diana and Sidney Woke Center for Memory Health at Hebrew Senior Life in Boston. I will tell you in a little bit about some of the work I do and why I do it. But let me start by telling you briefly about myself. I grew up in Spain, and I was interested in mathematics. I was interested in philosophy. I was interested in understanding what makes humans tick as humans. Why are we the way we are? But I grew up in a family of physicians, and my parents encouraged me to try to go at those questions through medicine. I went into medical school following their advice, and I went into medical school quite convinced I would devote myself to research and to understanding human biology, but not really going to be seeing a lot of patients. It didn't seem like something appealing. But when I started medical school, I found myself incredibly attracted by the care of patients, by the connection with the patients, by the empathy, by the opportunity to learn and try to help them, and by the joy of that role, in that honor that we're granted as physicians of helping others cope with their illnesses and disabilities. And so my world turned a little bit upside down, and I became a physician, and I would describe myself now as a clinician first, and motivated by my interest in helping patients. That is what really has continued to inform my research. So sometimes life is a series of serendipities, and in the setting of those serendipities, I've had the enormous pleasure and luck of meeting and learning from wonderful mentors, and it is their influence that has guided me along the way, and I am thankful for them. I have also had the opportunity of working with wonderful students and fellows and junior colleagues and colleagues. And so I will tell you about the work that I've done, some of the things that I have found particularly interesting and I hope interests you, and what I'm interested in and motivated to do now. But please remember that when I say me, I'm really talking about influences from many. My father, who passed away a couple of years ago, used to say that we are made of little pieces of lots of people. And in Spanish, somos hechura de todos. And I think he was right. And I certainly feel blessed by having been pieced together by influences of lots of people. So thank you again for the opportunity to talk with you. And I hope that you'll enjoy hearing about some of the work and experiments we've been doing.
You are an extraordinarily productive researcher with hundreds of publications and contributions. Of these, can you share some of your work that has attracted attention and why?
So I want to tell you a little bit about some of the experiments I've been doing over my career, some of the work that I find particularly interesting, and sort of share a few ideas with you that hopefully will trigger some questions and some conversations. I want to share my screen. I've put together a few slides, and so I hope that those illustrate my points better than my words might. So I'm again Alvaro Pascual Leone. These are my affiliations. What drives my work? My work is driven by the fact that about half of adults, of the adults over age 40, can expect to experience disability in their lives because of a brain-related disorder. I'm a neurologist, and I try to take care of patients with brain-related disorder. But most of us don't realize that brain dysfunction is a greater cause of disability than cancer and cardiovascular diseases combined. And that 1 in 3, maybe 1 in 4 people over the age of 65 will suffer from a neurodegenerative disease, including Alzheimer's disease. And so brain health should really be the number one target of overall health. And what I aim to do is help patients, one patient at a time, while gaining and advancing fundamental knowledge about the brain. In order to do that, I hope to contribute to empower individuals to define their life goals and to live their best lives. And in order to do that, really what we need to do, I think, is not wait until disease manifests itself, but aim to prevent disability, to prevent neurological disease. In other words, to sustain brain health. And so, again, brain health is what I'm after. Define it, maintain it, help people develop habits that promote their brain health goals. It is about a precision health. That's the goal. So the goal is a lifelong focus on brain health. And for that, I think we need a brain health index, a marker, a vital sign of how our brain is doing that we can use and measure across the lifespan. The rationale is as follows. Right now, we wait until symptoms manifest. Oftentimes, symptoms of disease take a long time to develop, and then we take time to go to see somebody to ask about them. But if we're lucky and find a good clinician, they will lead to a diagnosis, maybe after some tests, and hopefully be able to start a therapy. But oftentimes that is too late to really meaningfully impact disability. Talking in general about any diseases related to the nervous system, neurological or psychiatric. The fact is that before the symptoms ever show up, we know that the illness is there. Oftentimes, it can be there for many decades, as in the case of Alzheimer's disease. And at some point, we transition from health to having the disease, even though our brain was able to cope with it and we didn't develop symptoms quite yet. In fact, some people don't develop symptoms at all, despite having the disease, and that is true even for Alzheimer's disease. But most people will develop symptoms at some point, and it may be too late to really meaningfully help. So my focus is on trying to promote brain health and mental well-being, promote the capacity of the brain to sustain the illness and minimize the manifestation of symptoms, what we call brain resilience. And I think that is sort of a transformative approach that we're far from having as a standard, but that I think is where we should be focused on. In order to do that, I think we need to adopt a N of one symptom-based approach. We need to start with the patient. We need to define the symptoms rather than diagnostic entities. We need to understand what are the underlying physiology mechanisms that lead to those symptoms. We need to identify what goes wrong with those mechanisms that give rise to symptoms and then personalize the intervention. And we need to measure the impact while iteratively adjusting the intervention so that we get a best possible outcome. So the idea is to start with the patient and then reverse engineer the patient, including basic science approaches to bring back those insights to the patient for their benefit. If you start with the patient, then what we know over the life span of any one of us is that some people sustain function, we call them maintainers, and some people lose function, and we call them decliners. And for a long time, the assumption had been, even when I was studying, the assumption had been that the reason why people decline is because that's what age brings with it, and that it is unique individuals with a lot of, quote, reserve that are able to maintain function. But in the meantime, we now know that, in fact, we should, if we are healthy, maintain function, and that what drives us to not do so is the presence of pathology. So it is the pathology that makes us lose the ability to sustain function over the lifespan. Our abilities change. We become better at some things and we lose ability to do other things. But overall, we shouldn't decline. We should maintain function overall with some gains and some losses. So the need is there to keep checking, to keep measuring longitudinally over the lifespan. And the moment we start detecting very early a deviation, a decline, have that lead to the intervention and to seeing the physician, and not wait until much, much later when the symptoms are there to see the physician, as we often do these days. That's the goal of my overarching work right now. And I'll come back to that after showing you some experiments. But I wanted to frame this conversation in that context because it is promoted and driven and guided by that interest that I look at the research that we do to try to understand fundamental aspects of brain function, always with the goal of translating it to a benefit of individual patients and people. In that context, one of the things I've been most interested in is the notion that a healthy brain is a plastic brain. A healthy brain is a brain that sustains the ability to be modified, to adapt, to be changed by anything and everything we do. The ideas behind it stem from many, but particularly Ramon y Cajal formulated it by describing a number of abilities, including the job of a piano player, pianist, and he was arguing that is something that requires many years of mental and physical practice. It requires reinforcing established connections in the brain, making new connections in the brain through ramification and growth of dendrites. And so there is a complex process at play, but that process is critical, here my argument, to sustain a healthy lifespan and a healthy brain function. And I want to share one experiment we did a number of years ago around this topic. It had to do with actually doing what Cajal had told people as an example of that plasticity. It has to do with learning to play a 5-finger exercise on the piano. It was published a good number of years ago now, but I still like it and think of it as a particularly good illustration of these ideas of Cajal. So folks were seated at the piano. It's sort of rather boring, rather laborious. You have to learn to do 5-finger exercises up and down the scale, matching a metronome beat. And we connected the piano keyboard with the computer so that we knew exactly the sequence, the duration, the pressure, the interval between key pushes. I won't show you or belabor the behavioral component. People learn to do this. If you pay them enough, they will sit there 2 hours a day for 5 days and learn to do it. And of course, that's wonderful. But what we wanted to ask is the question, what happens in the parts of the brain, in the somatosensory and the motor cortex, that control those finger movements? And what happens, we could assess measuring the maps of cortical output, the projection, how much of the brain was connected to controlling finger movements. And in day one, in an area of about 1.5 by 1.5 inches over the motor cortex, you see a very concrete, circumscribed area that leads with a high degree of probability to finger movements when stimulated. And we stimulated it transcranially through the subject's skull. Over the course of multiple days, here is where that area is in the brain, but over the course of multiple days, that area grows and more of the brain is devoted to controlling the finger movements in a very rapid, very dramatic way. So that, again, over that brain area, after 5 days, a lot more of the brain appears devoted to fine finger movements of the piano. Now, remarkably, we showed that this expansion of the area that moves, that controls the finger movements, is related to the training and the learning. If you simply move the fingers, that's a control condition, but you don't actually learn to do the 5-finger exercise, that leads to some changes in the brain, but way less than if you actually acquire a skill of the piano playing. And if you don't move at all, nothing changes. So over the course of a week, as you're learning, this expansion takes place. And then we ask the question, well, so what happens if you keep practicing? What actually happens in the brain? Does the brain permanently change and become devoted to the hand? It wouldn't seem like a very adaptive thing. And in fact, what happens is that if you pause over the weekend and come back on Monday, there's been a shrinking of the representation. And then it grows again by the end of 5 more days of practice, only to shrink again and grow and shrink again and grow and shrink again and grow. But if you look week by week by week, you notice that over the course of multiple weeks, that growth from Monday to Friday becomes less, and that shrinking on Mondays becomes also less. In fact, there is 2 mechanisms at play. One very rapid expansion over the 5 days of practice each week, and then a much slower process that takes weeks or months of modification of the map in a more sustained and permanent way. And that maps onto the biology of it. There are changes of existing brain connections. The connections exist, but they're used more efficiently. And then there is a second process of actually making new connections. It takes time to develop, but is more sustained, is more consolidated, and is more lasting. So plasticity has 2 steps. The first one is influenced by the connections we already have and is very quick. The other one, by repeat and sustained effort, you can modify the brain and lead to structural changes. The remarkable thing about the experiment we did back then is that we found out that you could achieve the same by physical practice, actually playing the piano, or by mental practice. If you simply imagine that you were playing the piano but didn't actually move, you simply rehearsed it mentally, that mental rehearsal also changed the brain. And it's important to realize that this is one example of a few things that we now know about the brain. The brain is modified, is plastic, and is changed very rapidly, and it changes literally with everything we do, including what we think about. Therefore, thinking is a big responsibility. You better be careful what you think, because it will change your brain, and you may not like the consequences. Because the consequences of plasticity are a bit of a double-edged sword. There are beneficial consequences of plasticity. We call them adaptive, but there are also maladaptive mechanisms of plasticity or changes that result in symptoms of disease. A plastic brain is a healthy brain because a healthy brain needs plasticity, but the consequences of plasticity are not necessarily good or bad. They are simply the way that the brain functions by being modified. And the consequences of that modification can lead to disability and symptoms of disease, as well as to the acquisition of new skills. One example, again, to stay with the piano playing, is that if you play the wrong way, if you play with trauma to your fingers, or if you have certain genetic predispositions, then your representation of the fingers doesn't grow and maintain individual fingers, but rather grows and blurs the representation of the fingers. And when that happens, you develop dystonia, you develop alterations of motor control that ultimately cause cramps when you're trying to play, and that may render you disabled and ultimately unable to do the skill. So plasticity is the cause of symptoms, and it's the substrate for the acquisition of new skills. It is the fundamental way of function of a brain that is healthy and vibrant. We want to sustain plasticity, but we need to guide it. We need to guide the plastic changes so that they result in something that is beneficial for the individual. And I've devoted a lot of my time to study those mechanisms of brain plasticity, and I wanted to share that experiment as one illustration of the kind of work that I've been interested in doing. Now, one of the settings, one of the examples where we've explored plasticity and the degree of plasticity is in the setting of blindness. And again, the underlying core question is a fundamental question about the brain. Can we learn about seeing? How do we see? And then the translational aspect, the clinical aspect is, can we, by learning about this fundamental aspect of the brain, help individuals who lose sight cope with that in a better way? So my goal for a number of years was learning about seeing from the blind. Now, most of us think that the brain has sort of a bit of a camera kind of simile. We have the eye, we capture the world, it goes to this particular part of the brain, and we go, oh, I see X, I see something, some object. But in fact, that's not how the brain works. It turns out even more, if you look at brain imaging of people doing different tasks and look at that part of the brain that we call the visual cortex, in all these instances, the visual cortex is activated, but it turns out none of these people are seeing anything. They are reading Braille and are congenitally blind. They are localizing a sound in space and are congenitally blind. Or they are remembering verbal information and are congenitally blind. So it would seem that in the blind, the quote visual cortex doesn't do vision, but rather is engaged in something else. And you could say, well, that's because of this amazing plasticity in the brain that recruits parts of the brain that would normally be used for vision and now devotes them to do something else. But the work that we did actually leads to a different conclusion. Yes, the brain is plastic, but the brain is also organized in a different way than we thought it was. It is not the case that the brain is sort of a receptive camera where signals from the eye go to particular parts of the brain and we see, and signals from tactile information go to a particular part and we say, I read Braille, I touch. And signals from a different type go through the ears to a particular part of the brain and we hear something. It looks like it is the case, like that is the case, but in fact, we now have come to realize that our brain is generating a hypothesis and is then looking for the sensory system, for the sense that would be best suited to actually check on, contrast that hypothesis. So it's not a camera receiving information, it's a machinery generating hypotheses and looking out, going out to check on those world hypotheses with the external reality. And if you start thinking about it in that way, then you come and we propose an organization of the brain that we call a metamodal operator organization, where there are specific parts of the brain that have specific functions: detect shape of objects, discriminate precise spatial patterns, check on the timing between things, and so on. And then those operators, say a shape operator, goes out into the world using the senses to use the best signal possible in order to achieve that one thing it's trying to do. And if you start thinking along those lines, then it turns out that the apparent reality of the brain organizes a visual cortex, auditory cortex, tactile cortex, is really only an appearance, is the appearance generated by the fact that a given signal, say vision, is the best one for a particular thing, say spatial precise discrimination. But if you don't have vision, well, you're going to still try to do precise spatial discrimination, but you're going to do it by touch and dots of bumped braille. And the timing, you're still going to do timing discrimination, but not on sound bites. If you're deaf, you're going to do it on motion of American Sign Language, for example. And so your auditory cortex will be a visual cortex, but it's not really a visual cortex. It's still doing a temporal discrimination of events, just not in the sound domain, but in the visual domain. So that fundamental way of thinking of the brain as metamodal operators was one of the fundamental consequences that came out of these experiments of plasticity in the blind. And it has a lot of implications, not only about fundamental aspects of brain organization, but also one can translate it into strategies for teaching, rehabilitation of the blind, of the deaf, at the end of the day, of any and all of us. Now, I want to slightly switch gears, if I may.
Your work is patient-oriented. What's the translational relevance? And how can this improve patient treatment?
I'd like to tell you about another experiment and another aspect of particular importance in my work, and I think of translational relevance, but also fundamental insight in terms of how the brain works. And to do that, I want to share a slide, hopefully to illustrate the point and walk you through it. So the idea here is that brain functions are really the consequence of activity in distributed brain circuits. Distributed brain circuits that, as we've talked about, are plastic, and the symptoms of disease, the disabilities caused by dysfunctions of those plastic networks or circuits that we can call circuitopathies, are really the substrate of the neurological and psychiatric diseases. And in that context, the goal ultimately has to be for personalized treatment. We need to identify where is the problem in the brain to be able to go after that and ultimately decrease the disability of the patient. Notice that this is a little bit independent of what the cause of the dysfunction is. So the argument here that I'm trying to make is that the way I think about it is not that you have depression, you have Parkinson's, you have Alzheimer's disease, and we want the treatment for those. I'm making the argument those are too far away from the reality of the individual person. The patient is suffering from difficulty initiating movement, or from a lack of motivation to face life, or from a problem with autobiographical memory. And those symptoms, whether they are caused by one disease or by another, map onto dysfunctions of brain circuits. And if we can identify those, we can hopefully target those abnormalities and help the individual, while at the same time gaining insights on brain function at a fundamental level. These ideas are not new. In fact, Konstantin von Monachov, the turn of the century of the 18 to 1930s, argued that one needs to understand where a lesion is in the brain. That was necessary, but was not sufficient to explain the symptoms of a patient. That in fact, one needed to go from the lesion to understand the functional impact of that lesion onto the brain, to truly then and only then get to the point of understanding the symptom or disability. And I think we now have the approaches and the tools to do just that. So I want to illustrate that for you. And I want to illustrate that with a patient who presented with peduncular hallucinosis and was seen by Aaron Boyes, whom I'll tell you about a little bit more. And Aaron noticed that the patient was describing well-formed, vivid, elaborate, colorful visual hallucinations. She was seeing moving people, animals, well-defined, in sort of unusual settings. You don't see elephants in a room. They had sort of unusual environments, but well-formed, identifiable hallucinations. Turns out peduncular hallucinations was described by Lermite a long time ago, and many cases have been described. It's not that rare, and in fact, if you look at the literature as Aaron did, and I look at where the lesions have been that have caused this, in this particular patient is that little white dot in the thalamus of the patient. But if you look in the literature, you see lots of possible localizations giving rise to the same symptoms of peduncular hallucination. So again, like von Monachov said, lesion localization alone is not sufficient to explain the symptoms. It may be necessary, but it's not sufficient. What Aaron did with Mike Fox is to develop a method called lesion network overlap or lesion network mapping that give rise to a way to localize the functional impact of a lesion onto an individual's brain. And you can do that with any number of individual lesions. And when you do that, then instead of finding a very varied landscape of lesions in multiple locations giving rise apparently to symptoms, what you find with this lesion network mapping is that very many different lesions end up affecting the brain in such a way that very specific and concrete locations consistently end up being affected by remote impact of a lesion that can be in different locations, but has an impact on a consistent network, and with that, on a consistent target area. In this particular case, in blue, on the map, every single patient with this peduncular hallucination, regardless of where the lesion is, ends up having the function of what we call visual area MTV5 affected. And when you have that, then you have a localization for the symptoms, because that part of the brain has to do with motion, visual perception, and it's fed by information about the visual world and the objects in it. And so if it gets unleashed, it can give rise to the perception of visual forms, to visual hallucinations. Now we have a symptom localization, but with that, we do have a personalized target for the treatment, be it with specific interventions, brain stimulation or otherwise, be it with medications, if there are such, that can target selectively those areas identified in such a way. Aaron Boyes and Mike Fox and other colleagues have gone on to look at this approach of localizing dysfunctions in the brain and symptoms of disease with the lesion network mapping across multiple different types of presentations. But I think this illustrates the approach and highlights the importance of distributed networks and the pattern of activity in those networks as a fundamental building block to what we're able to do as humans and the symptoms of disease that come from alterations in them.
In what direction is the field going? What is the next big question that the field should be focusing on, and why?
Oftentimes, the field of neuroscience focuses on understanding the consequences of plasticity. How does the brain change because of plasticity when you lose vision? How does the brain change because of plasticity when you learn American Sign Language? How does it change the brain that you develop a given illness, and so on? And that's incredibly powerful and important. And I've shown you some experiments that I find illustrate important aspects. But at the end of the day, ideally, we would want to go beyond that to understand or to develop ways to measure how effective are the mechanisms of plasticity in a given individual. Because if it is true that the brain is intrinsically plastic, it has a capacity for plasticity, and that a healthy brain is one that sustains that plasticity, then it would be important to be able to measure and characterize and quantify the mechanisms of plasticity, because it's possible that they may be abnormal in themselves and that that abnormality may lead to disease and consequences. And so part of the work that we've been doing, that I've been involved in, has been focusing on trying to develop ways to characterize the mechanisms of plasticity and measure their efficacy. And I want to show you how that is done and why that is important. So if we want to assess the mechanisms of plasticity in humans, it turns out we can take the same approach that has been taken for some time to understand the mechanisms of plasticity in a slice preparation of the brain. We give a stimulus and measure response to it. In the case of the brain of a human, we can do it transcranially, non-surgically, using techniques like transcranial magnetic stimulation. And so we can use transcranial magnetic stimulation, record the brain activity, for example, with EEG or with EMG by evoking a twitch, and measure response, and we can define that as a baseline. Then, just like we would do in a slice preparation, we can apply a train of stimuli, a certain pattern of stimulation that will modify connections in the network that is being targeted. We can, after that train, measure again the responses. And if you apply the pattern in a certain intermittent rhythm, you get a potentiation of the responses. They're higher, larger than they were at baseline. If you apply a different pattern, you get a depression of the responses. We speak of long-term potentiation-like responses and long-term depression-like responses. Long-term potentiation and long-term depression are fundamental mechanisms of synaptic plasticity that have been described in a slice. We don't know in humans, in this form of application, whether what we're measuring is truly synaptic plasticity at work or is more complicated than that, and it's likely to be more complicated than that. But it behaves in a similar manner to LTP and LTD, hence the terminology of LTP-like and LTD-like. And importantly, one can measure it and characterize it in different parts of the brain, in healthy or patients with various diseases. One can look at the time course of that plasticity. One can characterize whether it is potentiated and abnormally so or insufficiently so, and the same for the depression. Now, we've known for a while, because Williams James, who coined the term plasticity, sort of made that argument from the offset, that there might be a critical amount of plasticity where it is good to have the right amount, but it's not good to have too little or too much. Williams James said that you want a system that yields to influence, that is able to adapt, but that doesn't yield all at once, hyperplasticity, excessive plasticity being potentially negative, but it is also not so resistant that it doesn't yield at all, hypoplasticity being abnormal. Well, it turns out we now have approaches to measure the amount of plasticity, to characterize population norms, to look at it across populations, to try to see if certain diseases might be linked to abnormal mechanisms of plasticity. And it turns out that is the case. It turns out that, for example, in Alzheimer's disease, very early on, we find a loss of LTP-like plasticity, that the system, the brain, becomes less plastic than it should be, And that might be the beginning of a whole cascade of problems that leads ultimately to the symptoms of dementia. On the other hand, we know that there are diseases like autism spectrum disorders that have an excessive amount of plasticity. They are hyperplastic, and that abnormality might be conducive, depending on the influence around the individual, to either becoming incredibly gifted in one thing, savant-like abilities, or learning, in quotes, so many things that interact with each other that the brain becomes noisy and they become disabled. So I think we now have the tools to characterize plasticity, and in turn, we have the building block with this type of measures to start looking at brain indices of health of the brain across the lifespan. And so I want to finish with the notion of where I'm trying to head towards, which is this identification of a vital sign of brain health that can be applied lifelong. And I want to give you 6 final ideas because I think they are important in approaching this type of thing. The first thing is that we need to remember that we need to focus on the whole person, that the brain doesn't walk along. The brain is part of the whole person, with an environment, with the surrounding, with the social reality, and that we need to capture those interactions as much as we need to focus on the brain per se. The second idea is that we need to focus on prevention. We cannot wait until the disease is present. We need to identify deviations from brain health and intervene there, including supporting the individuals and identifying lifestyles and lifestyle modifications as critical components of what determines brain function. The third aspect is that we really should embed these measures of brain function in everyday life, and we can leverage technology to do that. It doesn't mean that we need to take out clinicians. It just means that we need to extend clinicians using technology. It's not simply about empowering individuals. That is critical, but it's empowering individuals and connecting them with appropriate experts that can guide them. And we now have the technologies to take things outside laboratories and outside clinics and to do more real-life experiments in education, life settings, at people's homes. And I think we need to do that. When we do that, what we're going to do is 2 things at the end of the day. One is we're going to flatten the care system. We're going to make it much less discriminating to some folks. We're going to have a whole ecosystem of care that has individuals connected with the appropriate experts. And we're going to not simply measure the consequences of things, but actually, quote, look under the hood, capture brain function as it relates to behavior, for example. And with that, we're going to open up the possibility for a true precision, personalized approach to brain health. And I think that is not science fiction. I think that is possible. It may lead to a change in how we approach brain disciplines, brain study, neuroscience, education, and ultimately neurology and psychiatry. And I look forward to discussing some of these ideas with all of you. Thank you again very much for your invitation, and I hope that you enjoy my remarks and that trigger some discussion. And thank you.